Share screen. There's a lot of anatomy in the beginning, which will make this discussion very riveting, right? I think we can essentially breeze through, but Jasmine has volunteered to take figure 1, right?
Yeah.
Okay.
So, figure 1 as a whole, this shows the estimate of the INS to the PSTN area. A specifically shows a drawing that shows how the basal ganglia network is set up in the motor cortex to the STN, and the arrows depicts the 3 different pathways. The pink arrow would be direct, purple is indirect, and the blue would be the hyperdirect. For B and C, those are the fluorogold injections within the PSTN, but more specifically, B shows the labels of the cells in the 5th layer of the INS. C shows that the predominant amount of cells are in the ALD and the ALP. For D and E, B shows the drawings of the PHAL injection sites within the insular cortex. D shows the organizations of the projections, and in blue, it would be the STN, the PTSN, CBN would be in green, and the parva box would be in yellow, while E is showing how the isocortex innervates with the STN in the basal ganglia, hyperdirect pathway, and the INS innervates the PSTN, with the exception of the ALV, because that one would target that CBN and the parva box.
What's ALD and what's ALP?
That is a good question. Excellent
That's
question.
a lot of acronyms. Like
yeah, a lot of acronyms.
What's the INS? And why is it important to look at INS?
Man, no, I'll just look it up right now. INS is the INS the same as the insular cortex?
And what does especially for us, why is insular cortex so interesting?
Oh, the more we know, I'll look at that. Okay.
So wait, let's just see what they did in B.
Yeah.
What's the experiment in B?
In B,
yeah,
it was the fluorogold injections.
So where is fluorogold in what is fluorogold, or what does it do?
Okay. I'll search that up again. You know, the more we learn, fluorogold injection.
And anybody, I'm not just quizzing Jasmine, I'm just asking anybody to contribute.
It deals with the sensory nerves, and does it also have something to do with like damaged neurons?
Fluorogold? What is fluorogold used for? Anybody?
I think it's a retrograde tracer.
So what does that mean?
It gets taken up by the terminals where it's injected, and then goes back and labels their tracks of where the terminals come from.
Okay, good. So then in this experiment B, where's the injection in? Where are they injecting fluorogold?
Are they injecting it in like that PSTN area?
Okay, so in B, they show the site of injection, right?
Yeah.
And fluorogold, this one probably they did antibody staining for after fluorogold injection. And they're showing that they injected in the PSTN, but that's not really the purpose of the experiment, right?
Yeah.
The purpose is to see which brain regions send their projections to the PSTN. So then when you section the other regions of the brain and you see these brown stainings, you would conclude that the cells in that region are sending projections to the PSTN. Does that make sense now?
Yeah, that makes sense.
Okay, so then they probably sectioned the entire brain and looked at where the cells are, the brown cells are that send projections to the PSTN. Right, and an example of which is C, right, which is a cross section of the cortex in the insula cortex area, right?
Mhm.
I was just looking at something else. All right. So there are some cells there that send projections to the PSTN.
Yeah.
And D is a diagram that summarizes all the labelings that they saw. So they did this with the STN, they did this with the PSTN, and show that the projections to the PSTN are mainly these like green regions.
Yeah.
And then what is F?
So F would be like a dark field illumination after the immunohistochemical detection of the PHAL. And then G is also the same, but this time, like the difference between F and G is that it's the injection of PHAL into the motor cortex for F, but for G it would be the ALP, and for H it would be those injections in ALV, but that one's a photomicrograph.
And what does PHAL do?
That is an amazing question. Look it up. So let's see. PHAL.
What is PHAL? Anybody? Jake looks like he wants to say something, but he's holding back.
Yeah, go ahead and say it. You'll be helping me.
It's an anterograde tracer, so it goes the other way. But it's not a. I can't. Is it a virus or that? I don't know.
It's some like agglutinin or something, isn't it?
Okay.
Some glycoprotein. So that's an anterograde tracer. So now you're injecting in the insula and looking at where they're sending their projections to. So this is like the reverse of BNC. So they inject in the motor cortex and they see that there's a lot, there are fibers that are from the motor cortex that project to the STN. You see this region, right? Then when you inject in the ALP, then you see staining in the PSTN. Wait, this must be AIP. Isn't it agranular insula? Is that what AIP is?
Yeah, supposedly it is.
Yeah. Yeah, it's the agranular area of the insular cortex. So it's AI, and the probably dorsal, posterior. GU must be gustatory. what is this, visceral. These are just subdivisions of the insula. But the agranular dorsal, no, posterior AIP injection of PHAL shows projections in the PSTN. Yeah. So everything can be nicely summarized by this. The PHAL injection sites are shown by these red dots. What are these, like hexagons? No, heptagons? I don't even know how many sites are. But these things are where they made the injections in, and they see that they send their projections to the PSTN. Yeah. Anything else about this figure?
That's all I had.
Anyone else? All right. All right. Thank you, Jasmine. We'll go to figure number 2.
Yep, that's me.
All right. Great.
Spent a while on this, so we hope I cracked the code.
Mhm.
So we're trying to trace the overall pathway between the INS and the PSTN. And so 2A shows a general background of the corticostriatal projections from the cortex to the STN. And so it firstly shows that the INS targets AST, ACB, which is the nucleus accumbens, and FS in the CPU. They focus a lot on the AST, which is a ventral component of the CPU, which is the caudoputamen nucleus.
A lot of acronyms again.
I know, I found some of these acronyms though. INS also innervates the CEAC, so that's the caudal part of the CEA, and the CEAL, which is the lateral part.
And what's the
Additional-
CEA?
That's the one acronym I didn't find. I can find it for you though.
Does anyone know what CEA is? That's
Central nucleus of the amygdala.
okay. Good. So it's the amygdala, right?
Right.
That's the challenge of reading these papers. It's heavily anatomical because it takes a while to get familiar with the anatomy. Right? Otherwise, it becomes just like acronyms after acronyms.
Yeah.
A foreign
I had to go
language,
back.
basically.
Somebody was Alex was
Me, yeah, a foreign language, basically. I could not read this paper. It was impossible. I need like post-it notes on every page.
It's because the
Horrible.
French wrote this. Right. So
And so lastly, with 2A, they pointed out that the AIV and AIP sections demonstrate innervation of the olfactory tubercle, which is OT. So then that's just the background. So B and C were dual fluorescence to mark the overlap of PHAL and GCRP. And they wanted to see if the innervation of AST extended to the CEA below. And so B and C proved this to be true by providing evidence of PHAL labeled GU and VISC projections in the CEA.
So in B, what are they showing in B?
They're showing just the PHAL labeling.
Where was PHAL injected in?
I know I had a lot of trouble following. Oh, it's from the INS, isn't it? It was the axons.
Injection in the GU.
Okay. So we know where GU is, right? Here. the gustatory center. So that for people in this lab, that's interesting because that would be the taste cortex, right? So they inject in the GU.
Yeah, and so there were projections into the CEA of PHAL, which is due just from axons directly exiting the AST. So they show that the AST innervates the CEA essentially. And also from B and C, they noticed that the CEA innervation included the lateral portion, the CEAL, which is innervated by caudal AID and AIP, and the CEAM was not innervated by the rostral AID and AIP. And so from B and C, they concluded that INS innervation encompasses the lateral and caudal parts of the CEA.
Okay. So why do they even care about looking in the amygdala when this paper is supposed to be about the PSTN?
Because it's that indirect pathway.
Okay, good. So now they in this part they want to show that there's CEA to PSTN.
Yes, so they wanted to see if the CEA connects to the PSTN.
Okay, good.
So they did PHAL injections in the lateral CGRP rich regions of the CEA, and then combined this with FG injections in the PSTN, which I assume is retrograde. And they labeled cells that innervate the PST. The labeled cells that innervate the PSTN were lateral CEA regions, and then they also found like dually labeled cells in this SI region next to the global pallidus and a little in the CEAM, but not much. So they found in the SI region that the FG labeled cells are in close proximity to the PHAL axons. And so all of this shows evidence of a network between the PSTN or STN and projections from the INS, which parallel the direct pathway to the STN and indirect pathways through the CEA and SI adjacent to the global pallidus. And so G, H, I, and J are close-ups of the proximity of these FG cells to the PHAL axons. And that is what I got.
Oh, that's a mouthful.
I know.
But I hope everyone can follow the gist of the first two figures, right? The anatomy laying out the connectivity between the taste center, taste insular cortex, right, to the PSTN. Some directly, as shown in figure 1, right? Because when you inject retrograde in the PSTN, you see labeling in the insular cortex. And when you inject anterograde tracers in the insula, then you see staining directly in the PSTN. So that would be a direct connection, but then they also see this a circuit that goes through the central amygdala and other parts of the basal ganglia. No? Is that clear so far? Alex is not convinced.
This paper was not working for me at all. I cannot read this type of. I'm too old for this.
Mm-hmm. And it's because I think part of it is that even after you read this paper and all the, what is it, 7 figures, 8 figures, you really don't know what PSTN is doing. And I don't think the authors know what PSTN is doing. I think that's part of the challenge, that it's still in the early stages of understanding this brain region. So there's still a lot to be figured out that's out there. But also, as opposed to all the papers that we read previously, which was more sensory, like direct from the tongue. How does the tongue presentation of bitter solution on the tongue, how is that represented in the brainstem or other parts of the brain? Now we're dealing with something that is more, it's a higher order brain function, right? More of a forebrain function. So that makes it more complicated because there are more factors that go into that readout, and because they think that PSTN is involved in neophobia, they're looking at all these higher order brain regions. And they have to go through these basal ganglia and the central amygdala, the fear centers, or and motivation. So that makes the story already more complicated because these are complex behaviors. Right? So figure 3. It's more of the same, right? Tracing from the insula to the striatum. So I don't think we really need to delve into this still. In fact, the diagrams that they have in figure 1 and 2, especially in figure 1, is pretty helpful in just establishing that connection from the cortex to the PSTN. All right, so let's go to figure 4. So now they look at behavior, and this is where the more interesting aspects of this comes in. So can I have a volunteer for figure 4?
So here they are just trying to testing the role of PSTN in the test of neophobia and the sickness.
Mhm.
So because since I think previously they found there is a projection from the CGRP neurons input from the I think it's the PBN neurons. So that's they believe since the CGRP projection are involved in these two reactions, they want to see if PSTN is also involved. So there are two parts. First is they want to test how the neophobia triggers or activates these neurons, and they find that in panel A, if they have I think it's a rat, right? If they have a group of rats that are habituated to drinking the sucrose, and I think sucrose, right? Yes. And then they introduced this neophobia to the, oh, it's snowing outside. Okay. And then if they have another group of mice that have been drinking this water for the first time, they can see a significant difference in the signals. I think that is the neuronal reactions, right? And then in comparison for the mice that are habituated.
These are licks.
Oh.
Right?
Okay. Then, then the licking frequency is higher for the habituate control group. Right? Because it's denser. You can see that for it. Yeah, and for B, they also see the quantity changes, right? For the neophobia group, they have a much decreased drinking of the water. And in C, they're trying to test the CFOS expression, which indicates the neuronal activation of these PSTN neurons and also CBN neurons. They find no significant difference in the CBN. However, in the PSTN, they found I would say they found a relatively significant difference in the number of CFOS neurons. And in D and E, they further project these CFOS neurons to compare to the CGRP neurons. CGRP expression neurons, and they find an overlap here, indicating that these are the neurons they are targeting.
So if
Right.
if these PSTN neurons are involved in neophobia, which is what they're trying to suggest in panel C, why would the habituated neurons also show activity? Or in the habituated mice condition, why would the PSTN still show activity?
Well, probably they have some native expressions for the drinking behavior itself, or yeah, I mean, because CFOS is not like exclusively to neophobia itself. It can, I think, it can be related to many activities. So maybe here they're just focusing on the difference that's a number of increase in the CFOS neurons.
And this is a question for Jiahao, but Jiahao, what is this CBN? Do you see staining in the CBN?
It's based on the carbanding staining. They have a previous paper showing that, and just by the what they show in D and E, you cannot tell like where it is. The same for them. The definition just comes from immunostaining. Like there's a preferential carbanding. I think it's not carbanding, it's carbanding one. That basically means
CALB1.
Yeah, CALB1 expression specifically there, and it looks like a kind of like a nuclear-like thing, but
and the staining, the CGRP staining that they show here, is that the projections that are coming from the
it should come from the PD parabrachial nucleus.
Yeah.
What I feel they could do the show out like say, just like in figure 2, whether there is any suspected contact with the C4 cells by CGRP terminal. They didn't show, probably more like to have using the CGRP as a marker to show they are looking at the PSDN areas. And regard to the habituated animal, I think that's a good point, because this habituated animal just stopped drinking like after 15 minutes, especially in the example A.
Yeah.
So you might assume that you don't need the excitation of, to have the neuron to waste energy to make another, to start some new program making proteins, just in case, just because you taste again the same thing. This doesn't really make sense if in this aspect. So there must be some other things that signify it. It may be because the animal expecting those like say habituated sucrose, because those animals are water deprived. So it counts actually as them as a reward, they say. So they probably expecting this in that condition, like water deprived condition. So this kind of like signifies, some very positive experience with drinking in that setting, even though they are so much restricted. I think in that sense, PSTN probably have like populations that work for appetitive, and also populations that works for aversive, kind of like a response, taste responses. Those should come in a different, in a little bit different circuit. Let's say I probably assume that the CGRP shouldn't have like direct contact with those C4 cells expressed in the habituated animal, but have more direct contact with those C4s in the phobia animal. Just the hypothesis. I don't know.
And do you see with the P2X2 experiment, do you see in some CFOS in the CBN? I thought
probably not.
The PSTN region. Yeah.
Yeah.
Okay. So you sorry to interrupt. Figure F, panel F.
They are testing the sickness. So sickness can be induced by either LPS or cyst. So they did the IP injection into the rat, and then they find there are a significant increase in the number of CFOS cells in both the PSTN and the CBN this time. Yeah, so pretty much they are showing that these PSTN neurons are largely involved in sickness as well as neurophobia.
Yeah, I don't know what this means. So, based on just like this figure 4, but and where that comes from. And presumably the sickness information will come in from the parabrachial nucleus. The CGRP neurons that that gets activated in the parabrachial nucleus and send their projections to the PSTN.
Likely.
Somebody said something. No? All right.
This one.
Yeah.
So here they're switching to mouse, and so they wanted to first kind of check the anatomy and make sure it was similar to what happened in the rat. And so C is kind of just a diagram of overall what they did. They injected I think, anterograde tracer into the PSTN that was dependent on CRE expressed by the TAC1 mouse line. So similar to what we'll be doing soon. And so that labels specifically the PSTN area better than just injecting a normal tracer there. And so then they found, so yeah, A and B just show the injection site with the red is showing the center of the injection site. So you can see that it's pretty localized to that area. And then D through F are showing AAV and CGRP projections to the amygdala and SI areas that we saw in the rat, with green being projections from the PSTN. And then I guess one thing that was interesting here is that the CGRP innervated area doesn't get very much input from the PSTN. And that's those neurons that are presumably like the Cygnus neurons that are projecting there. Yeah, that's the main thing there. And then the next thing they did was they looked at CFOS from in G through I is from the neophobia, and J through L is from the Cygnus. And so you can see that there's overlap in the CFOS staining induced by the novel experience and with those TAC1 neurons from the AAV injection. And then similarly, but less convincingly with the LPS, there's overlap in CFOS staining there. And I think it's interesting that the CFOS staining especially the sickness one doesn't look like it overlaps with the TAC1 neurons that much, which...
Jay, can I interrupt you?
Yeah.
I didn't catch that. When going back to figure F, what
Yeah.
were you saying about CGRP? That what was the difference between this and the previous CGRP?
So I think there is less overlap between
that red region in the CA.
Yeah, and there was a lot of, I think in the rat there was a lot of overlap between the, if you see in figure D.
So this would be looking at projections that originate from the PSTN, right?
Yes.
And the projections from the PSTN seem to, in F at least, or ENF, you see that a lot of it is in the central amygdala M, right?
Yeah.
And not so much in the C or the L regions of the central amygdala.
Yes.
Right? And but in the previous figure 2, was it?
And figure 2, in like especially D, you can see it well. There's more overlap from those populations. So the rat seems slightly different.
In the D, that overlap would be coming from the insula cortex, right?
Okay, that makes more sense then.
And so these are all like slightly different. In figure 2, A to D, you're seeing how the gustatory cortex projects to the central amygdala. In 2, let me go back. So here, A to D is injecting in the insula and then looking locally in the central amygdala. Right? So all these projections, the green projections, are coming from the insula. And here you can see that the cell bodies that are in the CAM are sending their projections to the PSTN, right? Because they injected fluorogold, a retrograde tracer, into the PSTN, and they see staining in the CAM, which is this, right? But if we go back to, what is it, figure 5 that Jake was telling us about, they're injecting virus into the PSTN and looking at the terminals, and they see that there are projections in this region again. Not so much here, but mostly here. So it seems that the insula is sending information mostly to the CAC and the CAL. And then between the central amygdala and the PSTN, there's information going both ways, but mainly from the CAM to the PSTN, to and fro PSTN. Does that make sense?
Yes.
So here, yeah. So most of the projections that originate from the PSTN are in the medial region. Right. So let's go over the G to L again, because I was a little distracted and I didn't catch what you're saying.
Okay, yeah. So the G through L is looking at CFOS staining in the PSTN and how that colocalizes with the TAC1 neurons, basically. And so G is from the neophobia experiments, and there is a lot of overlap between CFOS staining and those TAC1 neurons. And they also say that there's a lot of overlap with the in J through L with the LPS injections, but I think it's a lot less convincing how much overlap there is there.
Mm-hmm. I mean, even G to I, there are some non-green red cells.
Right. Yeah, I was saying, I think that kind of makes sense with what Jahao was saying, with there are probably different populations of cells in this region doing different things.
But based on this, I'm not so sold on TAC1 being a good driver.
Yeah, that.
I mean, there's partial overlap and partial non-overlap. So. What do you think, Jahao?
It's not perfect.
Yeah.
I try to reconcile this non-overlap, like say partial overlap, and there's obviously a huge amount of cells C4 cell cannot be accounted by the TAC1 expression. Then when you proceed to the next figure and see what the effects of lockdown these TAC1 populations.
Yeah,
I think the effects is surprising if I combine those two.
Yes,
just like
but based on figure 5, and let's just, probably wrong, but assume that every neuron in PSTN is doing the same thing.
Okay.
Yeah, probably not true. They're probably very heterogeneous, but if PSTN is just doing one thing, then the effect of, let's say, removing or silencing the entire PSTN should be much stronger than silencing just the TAC1 neurons. Which I'm puzzled why they wouldn't do. Because especially for this, like there's very little overlap for L. What do you think?
Yeah, there's very little. So if they start with this LPS and CFOS in the TAC1, I think it will be hard for anyone just jump to the functional study.
Yeah,
like knockdown or even enhance the function to see whether there's a role in the LPS induced illness. It will be, it's somehow not supported. So it's probably started the other way around. Start with the whether the any population in the PSTN interfered with the LPS induced illness, and starting from there, and they find it does, and then goes back to see, okay, whether there is any indication that the TAC1 really works there. C4 is one of the ways to see the activity somehow previously have been happened. We have the photometry, which is more direct.
So we based on this, I don't know how good of a driver TAC1 will be, but we also have to remember that G to I is neophobia, but it's presentation of sucrose, right?
20% of sucrose.
Yeah, so I would think that 20% is pretty sweet. And so if some of these were taste dependent or tastant, not even sweet, but just some taste information, then that G to I seems to show more overlap than the LPS. So maybe for the taste part, TAC1 is not too bad.
So you mean LPS doesn't tap into the taste system? or?
Yeah, it shouldn't, right?
Okay. And your comment on the 20%,
Yeah.
their previous paper, like 5 years, like
Yeah.
2015, they compare how much, compare like 5% of sucrose, 20% sucrose among other tastings to rats, and they look at how much, like say, Stifol's expression they got from in the PSTN, and is strongest in the 20% of sucrose, not in the 5%. But behaviorally, the rats prefer the 5% of sucrose versus 20% in their assay.
How about we figure out what 20% sucrose means in molar concentration?
Yeah, I think it's probably it's a little bit not purely sweet taste for 20% now. Then it's probably something.
Does everyone know how to do this? To convert 20% sucrose to a molar concentration? Who knows how to do this? Who's confident that they can do this? Nobody? I guess some of you are being humble. But how would you do this? How would you calculate the molar concentration of something from percent weight per volume? No idea.
Let me try first. So you have the 20%. You roughly equal to, just take one simple number, like 200 grams in 1 liter. And then 200 grams.
That's right.
And we presume that just imagine there is like 1 liter of 20% of sucrose solution, and 20% of them are sucrose.
I don't want you to jump too fast because you'll lose everybody.
And then 20% meaning the volume of 20%.
Yeah.
So assume that the volume, then that means like 200. That means 200 milliliters meals of those volume are sucrose. And suppose the density equal to 1, which is should be greater for sucrose solution.
Jahao, that's great, but you're losing everybody because I don't think Jasmine, who's the youngest here, I'm not picking on you, I'm just pointing it out, but, would have no idea what you're talking about when you say density.
Okay.
Although it's important, right? But generally, we when we do weight per volume, we don't even consider it density. But easy way to think of 20% sucrose is how much, so gram per 100 milliliters, right? So 20% will be 20 grams in 100 mLs. Can everyone see the rough drawing that I'm trying to make on this? 100 milliliters. Does that look like 20%? Yeah, easily 20%, right? So that's what Jao was saying earlier, 200 grams per liter, right? So that this would be 200 grams per liter. Yeah. So now what's the information that we need to convert this to molar concentration?
Molar mass.
Okay, so what's the molar mass of sucrose? Is it like 300 something?
I just looked it up. It's 342.3.
0.3.
342 grams. So 342 grams in a liter would be what? 1 mole, right? 1 molar, right? So then you just have to convert, see what this would make. So that's pretty high. It's like 600 millimolar or something. Is that right?
Mm-hmm.
So that'll be, so we, if you do like 500 millimolar, I think that would be like the maximal taste response you get, like nerve recording or calcium imaging. So these are significant, very high, very sweet things, 20%. So Amanda or Jasmine, what's the molar concentration of this, of 20%? How do you go from here with these two lines? How do you convert that to
It
molar?
would be 342 grams per mole. And then you would do dimensional analysis to calculate moles.
Yeah, so what
And
is,
then you have the liter, so then you could find the molarity.
okay, so then what is it then? You just have to do like,
Oh, I'll grab a piece of paper.
200 for 300 equals x to 1, right? 342 to be precise. Anyways, that's how you do it. So it's not nothing complicated. And I actually hate it when people people put like percent in a paper.
Because that's easier to prepare the solution. So easy. You just weigh the sucrose, 20 gram.
Oh, I know we use 30% sucrose to cryoprotect our samples, but still. Okay, so get off of this. Okay. Oh wow, that stays there. Not know that. So are we done with this one? Oh, where are we now? A little bit lost. Here, so in G2I, they're presenting what, like 600 millimolar sucrose, 20% sucrose. So if some of these are activated by taste stimuli, just have to keep that in the back of our heads that this could be like a sucrose or a taste dependent activity, the G2I. And that would be conveyed by some of it will be by TAC1 expressing cells. Yeah. So if Jahao took a TAC1 Cree mouse and did photophotometry here, like he showed us on Tuesday, and the mouse is eating, drinking something sweet, 20% sucrose, then he might see some activity. Yeah. All right, so let's move on to, wow, we're past 1 o'clock. How did that happen? I thought we were going pretty fast. It's usually a bad sign when you guys make me talk too much. We want to breeze through these figures. All right, let's quickly go through figure 1. Figure 6. This one figure. which I think leaves Jiahao.
Me? I
Alex?
cannot see my screen because my screen was blocked.
That's a good excuse. Maybe Alex can go.
Alex can be a can be can be my witness. My screen is blocked.
Right. So what's happening in figure? This is the one. Oh, why did that happen? So this is the one functional experiment that they do. They're using threads, GI, to block the activity of TAC1 neurons in the PSCN. Yeah. And so they have the controls, two controls and the thread. And they don't see effects with the forced swim test. They don't see effects of open field. They don't see differences in the food intake. The two differences that they do see, one is that when you block the activity of PSTN, TAC neurons, then they see increased licks after inducing sickness, which to me is puzzling because not many of the TAC1 neurons were overlapping, right? In figure 5, J to L, there's not much overlap between TAC1 and the LPS neurons, but here it seems that when you block TAC1 neurons, you're getting more licking. And then there's this very small difference in my mind between neophobia in the neophobia condition. So when you block TAC1 neurons again in the PSTN, they're getting more licking and less neophobia. So in short, that means that when you don't have the activity of TAC1 neurons, again, it's not just the entire PSTN, it's TAC1, they see that those mice will approach new foods more, and consume them sooner than in control animals, and also that these animals will show less less aversion to licking when they feel sickness. Again, I think the data itself is quite weak here. And I think part of it could be just the TAC1 line that they're using. And if we just remind ourselves of Tuesday, there seem to be many things happening, right? The activity that Jiahao sees happen when food is presented, when zoo animal, a tasty cracker is presented, but also Eppendorf tubes cause some kind of a short burst, right? There was activity during, so that short burst went away with Eppendorf tube, but for food, there was sustained activity. So again, I think we are looking at multiple things happening, and we'll have to do these kind of experiments to figure out what's happening. Anything else about the paper? So Jake, during your rotation, I hope that we can figure out what's going on in the PSTN. And this is the one paper that shows perhaps a little bit of the function of the PSTN. This came out in July of last year. So there's very little known about this region. A lot of work to be done, and I think it's quite interesting. I don't know how all this ties into the insula innervating directly to the PSTN or the central amygdala directly to the PSTN, but those are things that we need to figure out. Any other questions? So, Alex, does it seem like we are still meeting on Fridays for a journal club?
Maybe, let me take another look.
Is everyone here good with Friday noon? Yeah.
Actually, Jasmine, Jahao, Jay, and Molly said that Friday noon is not.
It's not good for
not
Jasmine?
comfortable. No. It seems like Thursday and Tuesday 11 a.m. is comfortable for everybody except for GIE. And that makes sense.
Yeah. Okay, so then we might have to move the lab meeting to like a Tuesday 11, and then journal club to Thursday 11, which breaks up the day, and it's not ideal. Yeah. Okay. So are there more people that need to do the when to meet thing? Or
I don't think so.
okay, all right. So I'll take a look at it. We'll see what time is good. Personally, I don't think it's good to break up like the entire week like this, like the middle of the day. So if we can move at least one to an earlier or later time, I think that'll be better. But we'll figure it out. Okay. And Jake, are you coming in on Monday?
I should be, yes.
Okay. Well, Monday your classes start also, right?
Yeah, but we only have, well, I should make sure, but I think it's only 9 to 10 on Monday.
Okay, all right. So I might see you like Monday afternoon then.
Yeah.
Okay. All right. Enjoy the snow. It's like, wow, it's a lot of snow.
Yes, but it should stop soon. On the other hand, it didn't say it's going to snow today at all. So, yay, Evanston.
Oh, is that right? Wow.
Yes, it wasn't supposed to snow today. I wore the wrong shoes.
Jasmine, you don't get any snow there. Yeah. All right. Well, have a nice weekend. I'll see you all on Monday.
Have a nice weekend.
Bye.
Au revoir.
Hello. About the
Yeah,
email. Yeah.
I wanted to. I'm also recording, so I want to turn that. Also.